Today, we're going to review the structure of proteins. The monomer for a protein is an amino acid. Amino acids are organic. That means they are hydrocarbons. So the structure of an amino acid starts with a carbon bonded to a hydrogen, and then there are two functional groups attached. An amino group, which is an N bonded to two hydrogens, and a carboxyl group. The fourth place on the carbon has a variable R group. Monomers join with other monomers to form polymers. So that means that this amino acid is going to form a bond with another amino acid. When two monomers form a bond, the type of chemical reaction that takes place is called a dehydration synthesis reaction. Dehydration because we lose a molecule of water in the process, and synthesis because the two monomers are joined together. The type of bond that joins two amino acids together is called a peptide bond. What we have on the table are a bunch of individual amino acid monomers. Today, we're going to build the protein rhodopsin. We're going to start by joining these two amino acid monomers together. And we're going to use a piece of tape to connect the two monomers, representing a peptide bond. We're going to join all of these individual amino acid monomers together. So now we have the backbone for the amino acid chain, but we can't forget about those R groups. Remember that there are four categories of R groups: nonpolar, polar acidic, polar basic, and polar uncharged. We're going to use Post-it notes to represent these four broad categories. So for every R group that is nonpolar, we're going to use a pink Post-it note. And for every polar amino acid R group, we're going to use a blue Post-it note. We're going to use orange to represent the acidic R groups and yellow to represent the basic R groups. So here we have our backbone. Every place there is an R, I'm going to look at the type of R group that is attached for this particular amino acid chain, and I'm going to use Post-it notes to represent the type of R group. This is called the primary structure of the amino acid. It is the long, linear chain of amino acid monomers linked together. But remember that proteins have to fold in order to be functional. First, they fold by hydrogen bonding of the backbone into their secondary structure. There are two characteristic shapes, an alpha helix and a beta pleated sheet. This particular chain is going to fold into an alpha helix. So we are going to be folding this into what looks like a tube. And we're going to use hydrogen bonding of the backbone in order to fold and make this tube. Remember that hydrogen bonds are between oxygen and hydrogen. So we are going to connect the oxygen from the carboxy groups to the hydrogens of the amino groups. And we're going to use a piece of tape to represent the hydrogen bond holding those two together. So we're going to continue to twist this amino acid chain, matching hydrogens to oxygens. Once we're done folding, we have this alpha helix where we can see oxygens hydrogen bonded to hydrogens all along the backbone of this amino acid chain. This is a completed alpha helix with the R groups sticking out. The protein that we're building is not just made of one alpha helix. It's actually made of 7. And the R groups that are sticking off of this amino acid backbone are going to interact with one another to form the final tertiary structure of this protein. You can see in this picture the 7 alpha helices that make up this complete Bacteriorhodopsin protein.